All right, welcome back to the deep dive. Today we're going to be talking about leukocytes. It's, as a medical laboratory scientist, you're dealing with these cells day in and day out.
Absolutely.
But we wanted to kind of take a fresh look at their entire life cycle, really get into where do they come from? What are they doing? What are their different jobs? And then ultimately, how are they cleared out?
Yeah, we really wanted to go through all the different sources that we found
Right.
for this deep dive and really try to get into the nitty-gritty of leukopoiesis,
Yeah.
their function, both innate and adaptive immunity, and then how they're destroyed.
And this isn't just, reviewing a textbook. We really want to think about how does this actually relate back
Oh,
to
absolutely.
what you're doing in the lab.
Yeah, I mean, this is everything from, how you view a CBC to a bone marrow aspirate and everything in between.
Right, exactly. So let's get into it. So starting with the very beginning, leukopoiesis, it all starts in the bone marrow, right, with
Mhm.
the hematopoietic stem cells.
That's right. So the HSCs are really the pluripotent stem cells that give rise to all of our blood cells. And then uh, for our purposes today, those differentiate into the common myeloid progenitor, or CMP,
Okay.
and the common lymphoid progenitor, the CLP. And that CMP is going to give rise to all of your granulocytes, your neutrophils, eosinophils, basophils, as well as monocytes, which are the precursors to your macrophages.
Got it.
And then the CLP, of course, is what gives rise to your lymphocytes, your T cells, B cells, and your natural killer cells.
Okay, so let's get into the myeloid side of things. Um, let's talk about the granulocytes and how they're made.
Okay.
Granulopoiesis.
Granulopoiesis. So it's really an interesting process. It's a very regulated process. It all starts with that myeloblast, which is a relatively undifferentiated cell. It has a large nucleus, not a lot of cytoplasm,
Okay.
and then it matures into a promyelocyte, which is when we start to see those primary granules.
Uh-huh.
And those granules are packed with things like myeloperoxidase, lysozyme, all those kind of enzymes. They're important for antimicrobial activity.
Yeah.
And then from the promyelocyte, it matures further into a myelocyte.
Okay.
And that's when it gets really interesting, because that's where those secondary or specific granules start to develop.
Okay.
And that's really what dictates whether it's going to go down the neutrophil lineage,
Got it.
the eosinophil lineage or the basophil lineage.
Mm.
So at that point, you'll see neutrophilic myelocytes that have very pale pink granules. You'll see eosinophilic myelocytes. They have those striking orange-red granules.
Right.
And then, of course, your basophilic myelocytes that have those very dark blue granules. And actually, that's kind of referred to as the dawn of neutrophilia, when those secondary granules.
Dawn of neutrophilia.
Dawn of neutrophilia.
That's a cool
It is. a
little
little-
term there.
Yeah, I mean, really, at that point, that's when that cell is committed to becoming a neutrophil.
Its fate is sealed. All right, so what happens after the myelocyte?
So after the myelocyte, you get a metamyelocyte, and that's when you start to see that nucleus start to indent. So it kind of
Yeah.
gets that kidney bean shape.
Yeah.
And then it goes on to become a band form.
Okay.
And the band, the nucleus, is much more elongated.
Yeah.
It has that very characteristic horseshoe shape.
Classic horseshoe.
And then from there, it becomes a mature granulocyte.
All right, so then we've got our three mature granulocytes, right? Neutrophil, eosinophil, and basophil. And we can tell them apart on a smear.
Absolutely, yeah. I mean, each of those has its very characteristic morphology on the peripheral smear.
So what am I looking at?
So the neutrophil, of course, has that segmented nucleus, the multi-lobed nucleus.
Yeah.
And it has very fine granules that are neutral staining, so hence the name neutrophil.
Neutrophil, okay.
The eosinophil has a bilobe nucleus,
Okay.
usually 2 lobes, and very large, striking orange-red granules.
Right, yeah.
And then the basophil have a lobulated nucleus, but a lot of times it's really hard to even see the nucleus because those granules are so intensely dark blue to purple.
Right, they just stain so darkly
Absolutely.
that they kind of cover everything up.
Yeah. Okay, cool. So we've got the granulocytes down. So then on the myeloid side, we also have monocytes.
Yes.
So how does monocytopoiesis work?
So it's a very similar kind of developmental pathway.
Okay.
You start with a monoblast.
Good.
It matures into a premonocyte. The premonocyte's a little bit larger, has a little bit of an indented nucleus, and then some azurophilic granules. And then from there, it goes on to become a monocyte.
Yeah.
And the monocyte has that very characteristic horseshoe or kidney bean-shaped nucleus.
Yeah.
And it's got a cytoplasm that has fine granules and often some vacuoles.
Okay.
And I think what's important to remember is that circulating monocyte is really a precursor cell,
Okay.
because once it leaves the circulation and gets into the tissues, that's when it really becomes a macrophage.
Okay.
And depending on where it goes in the tissues, it takes on a very specific function.
So it's like going to boot camp
Exactly.
and then being deployed.
Exactly.
Okay, cool. Yeah, like the Kupffer cells in the liver.
Right.
Right, or the alveolar macrophages in the lung. They all come from that same monocyte precursor, but they become very specialized depending on their location.
Right. Okay, very cool. So that's the myeloid side of things. So now let's switch gears and talk about the lymphoid lineage.
Okay.
So we've got our T cells, B cells, NK cells.
Yeah.
How are they produced?
So lymphopoiesis also starts with a common lymphoid progenitor, or the CLP, in the bone marrow.
Okay.
And that CLP then differentiates through stages, including the lymphoblast and the prolymphocyte.
Yeah.
But from there, the maturation pathways diverge quite a bit.
Okay.
So D cells, they largely complete their maturation within the bone marrow.
Okay.
And that's where they develop those unique B cell receptors.
Right.
But T cells, they actually undergo a really important developmental journey to the thymus.
Okay.
And that's where they undergo this really rigorous selection process.
Yeah.
Where they make sure that those T cells are self-tolerant and that they develop their T cell receptors properly.
Right.
And then NK cells, or natural killer cells, they develop as large granular lymphocytes. And their maturation is actually a little bit more complex. It occurs in various sites, including the bone marrow, but also secondary lymphoid tissues.
So that's a big difference then, like if you're looking at a bone marrow sample versus, say, a thymus sample. Like,
Absolutely, yeah.
yeah.
The cellularity and the composition is going to be very different between those two tissues just because of that distinct maturation path for the T cells.
Got it. Okay. So clearly it's a very, very tightly regulated process to make all these different types of cells.
Yeah.
What are some of the things that are responsible for making sure that you're making the right number of cells at the right time?
So there's a whole host of different signaling molecules and factors that play a role in regulating leukopoiesis. One of the key players are the colony-stimulating factors, or CSFs.
Okay.
So you've got things like GM-CSF, which acts very broadly on both granulocytes and macrophages. G-CSF, which is more specific and really drives neutrophil production. And then M-CSF, which is important for the monocyte macrophage lineage. And then you've also got the interleukins.
Right.
So IL-3 acts on some of those early hematopoietic progenitors. IL-5 plays a role in eosinophil development.
Okay.
And then IL-7 is very important for lymphocyte survival and proliferation.
Got it.
So you've got all these CSFs and interleukins working together. And then you've got other factors like TNF and PGF-beta that can either stimulate or inhibit leukopoiesis, depending on the context.
Right, right.
And then within the cell, you also have these very important transcription factors.
Yeah.
Things like PU.1, GATA1, and Icaros.
Okay.
And these really act as master regulators that guide the differentiation pathways within the cell.
So what happens if something goes wrong? I mean, clearly this is a very finely tuned system.
Absolutely.
So if something is out of whack, what happens to these cells?
So when leukopoiesis is disrupted, you can end up with a whole spectrum of hematologic conditions.
Right.
So if you don't have enough production, you end up with leukopenia, which can manifest as neutropenia, which is a decrease in neutrophils.
Yeah.
And that leaves the body very vulnerable to bacterial and fungal infections.
Right.
You can also have lymphopenia, which is a decrease in lymphocytes.
Right.
And that can increase your susceptibility to viral infections.
Got it.
But then, on the other hand, you can have too much production, so you end up with leukocytosis.
Oh, okay.
And that's where you have elevated numbers of certain cell types. And that could be something like neutrophilia, where you have an increase in neutrophils. And that's very common in things like bacterial infections. You could have lymphocytosis, an increase in lymphocytes, which you might see in certain viral infections or in lymphoproliferative disorders.
Okay.
And then in the most severe cases, if you have completely uncontrolled proliferation of leukocytes, and often that's abnormal proliferation, that's when you end up with leukemia.
Right, right.
Which I'm sure you're very familiar with in the lab.
Exactly. Yeah, unfortunately, we see our fair share of those. All right, so we've talked about how these cells are made. Let's talk about what they do, right? Why do we even have them in the first place? So they're there to protect us.
Yes.
And their functions can be broadly categorized as innate or adaptive immunity, right?
That's right. So you've got your innate immune system, which is that rapid first line of defense against anything that comes into the body that shouldn't be there.
Right.
And that's where you see your granulocytes, your neutrophils, eosinophils, basophils,
Yeah.
your monocytes and macrophages, and your NK cells.
Okay.
And then you've got your adaptive immune system, which is a little bit more specialized. It develops over time, and it really involves your lymphocytes, your T cells and B cells.
Okay, so let's talk about each of those cell types
Okay.
and what they do, starting with the neutrophil. You know, we always think of neutrophils as those first responders.
Absolutely. Yeah, they're the first ones on the scene. They're really the workhorse of the innate immune system,
Especially for bacterial infections.
especially for bacterial and fungal infections.
Yeah.
Yeah. I mean, their primary mechanism is phagocytosis.
Right.
They engulf and destroy microorganisms.
Yeah.
They've got this impressive array of granules that contain all these antimicrobial substances, lysozyme, myeloperoxidase, defensins, elastase,
Right.
and they can release those through degranulation,
Okay.
which just means they're releasing the contents of those granules.
Okay.
And that can directly kill pathogens, it can break down tissue.
Right.
And then they also have something called the oxidative burst,
Okay.
which is basically they produce a lot of reactive oxygen species, and that's generated by enzymes like NADPH oxidase and myeloperoxidase.
Okay.
And that basically creates a very toxic environment within that phagolysosome
Yeah.
and helps to kill those microbes.
Right. Right.
And then they have these really interesting structures called neutrophil extracellular traps, or nets.
Oh, yeah. Nets.
And that's basically where they expel this web-like structure composed of DNA and antimicrobial proteins, and it actually can trap and kill bacteria extracellularly.
It's really cool how they can kind of shoot out those nets
It is.
to capture things.
Yeah, it's pretty amazing.
And then also, we should mention, they also have to get to the site of infection, right?
Absolutely. Yeah, that's chemotaxis. They're
Mhm.
able to follow chemical signals that are released at the site of infection, and that helps to recruit them very quickly.
Yeah, so they're like little bloodhounds.
Exactly.
Sniffing out where they need to go. Okay, cool. So that's the neutrophil.
Yeah.
So then let's talk about the eosinophil. We often think of eosinophils with parasitic infections and allergies.
Right. So they're key players in defending against parasitic infections. They release a bunch of different cytotoxic granular proteins.
Okay.
So you've got major basic protein, or MBP,
Right.
eosinophil peroxidase EPO, eosinophil cationic protein ECP, and eosinophil-derived neurotoxin EDN.
Okay, so all these different proteins
Yes.
that are toxic to parasites.
Yes, exactly.
Got it.
They can also phagocytose antigen antibody complexes, so they help to clear those out of the system. And then, interestingly, they also participate in modulating allergic reactions.
Okay.
So they can release enzymes like histaminase and aryl sulfatase,
Right.
which actually degrade histamine and leukotrienes.
Right. So they're kind of dampening down that allergic response.
Exactly.
Okay, got it. And then we've got the basophil, which is often implicated as well in allergic responses.
Yes, absolutely.
Mm-hmm.
So the basophil granules are packed with histamine, heparin, and leukotrienes.
Right.
And when they're activated, they release those, and that leads to the very rapid development of all those allergy symptoms.
Right. All that swelling, redness, itching.
Absolutely, yeah. Increased vascular permeability, smooth muscle contraction, mucus production, all of those things.
Right.
And basophils, they have these high-affinity IgE receptors on their surface.
Okay.
And when those receptors bind IgE antibodies that are specific to a certain allergen,
Right.
that triggers a cascade of events within the cell that leads to degranulation and the release of all those pro-inflammatory mediators.
Mm-hmm.
So that's really what's driving that very rapid allergic response.
Right. It's like a chain reaction.
It is. It is.
All right. So then we talked about monocytes already and how they're made, but let's talk about what they actually do.
Right. So monocytes, they're circulating in the blood, and they do perform some phagocytosis.
Okay.
But they're also really important for producing pro-inflammatory cytokines.
Okay.
So things like TNF-alpha, IL-1, IL-6.
Right.
And those really help to kind of orchestrate the broader immune response.
Okay.
But their real strength, I think, comes when they differentiate into macrophages in the tissues.
Okay.
So macrophages are just these really amazing cells. They're very efficient phagocytes.
Yeah.
They can engulf pathogens, cellular debris, tumor cells.
Right.
They also function as antigen-presenting cells,
Okay.
which is really important because that's how they link the innate immune system to the adaptive immune system.
Got it.
So they take up an antigen, they process it, they're presented on their surface to T cells, and that helps to activate the T cells.
Right, right.
And then, of course, we talked about how they can specialize in different tissues.
Right.
So you have your Kupffer cells in the liver, your alveolar macrophages in the lungs.
Yeah.
They all have these really specific functions depending on where they are.
Right.
And then I think one other thing to mention is macrophage polarization.
Okay.
So macrophages can actually become these functionally distinct
Oh,
populations.
so.
So you have your classically activated M1 macrophages, which are very pro-inflammatory.
Right.
And then you have your alternatively activated M2 macrophages, which are more involved in things like tissue repair.
Okay.
And that balance between the M1 and M2 phenotypes
Yeah.
is really important in a lot of different disease states,
Right.
things like cancer and chronic inflammation.
Okay, very cool. So that's the monocytes and macrophages. So then on the lymphoid side of things, we've got our T cells, B cells, and NK cells.
Yes.
And NK cells, while they're technically lymphocytes, they act a little bit more like the innate immune system, right?
They're kind of that bridge between the two.
Yeah, yeah. So let's talk about T cells first. So we've got helper T cells and cytotoxic T
Right.
cells.
So T cells are essential for adaptive immunity.
Okay.
They have these T cell receptors that recognize very specific peptide antigens.
Right.
And those antigens have to be presented on MHC molecules.
Right. So it's a very specific interaction.
Very specific. So your CD4 plus helper T cells, they're really the orchestrators of the adaptive immune response.
Okay.
They help to activate other immune cells.
Got it.
And when they're activated, they differentiate into all these different effector subtypes.
Okay.
So you have Th1 cells, Th2 cells, Th17 cells, Treg cells.
Right. All these different flavors of T cells.
Yes, and they all have these distinct cytokine profiles.
Yeah.
And those cytokines really shape the type of immune response that you get.
Right. Right.
So for instance, Th1 cells, they're very important for cell-mediated immunity against intracellular pathogens. Th2 cells help to support humoral immunity, so antibody production against extracellular parasites.
Got
And they're also often implicated in allergic responses.
it. Okay.
And then you've got your CD8 plus cytotoxic T cells.
Right.
And they're really the killers of the adaptive immune system.
Right. They're the assassins.
They are.
They go after specific cells.
They do. So they recognize antigens that are presented on MHC class I molecules. And MHC class I molecules are found on all nucleated cells in the body.
Right.
So they're constantly surveying, looking for cells that are expressing something that they shouldn't be.
Got it.
Whether it's a viral protein or maybe something that's indicative of tumor.
Yeah.
And when they find that cell, they basically kill it by releasing these cytotoxic granules.
Okay.
And those granules contain perforins and granzymes.
Right.
And those induce apoptosis in that target cell.
So it's a very controlled way of eliminating those cells.
It is. It is.
Okay, so that's the T cells.
Yeah.
And then we've got the B cells,
Yeah.
which are the antibody factories.
Absolutely.
Yeah.
Yeah, they're really the workhorses of humoral immunity. They produce antibodies, and antibodies are just these amazing proteins that can do so many different things.
Right.
They can neutralize pathogens.
Yeah.
They can opsonize them for phagocytosis.
Okay.
They can activate the complement system to directly lyse pathogens.
Wow.
And they can mediate something called antibody-dependent cell-mediated cytotoxicity, or ADCC,
Oh, okay.
where they actually recruit NK cells to kill a target cell.
Got it.
So each B cell has a very unique B cell receptor on its surface,
Okay.
and that allows it to recognize a very specific antigen.
Right. Right.
And when it encounters that antigen and it gets help from a T helper cell,
Yeah.
it can differentiate into a plasma cell, which is a very specialized antibody secreting factory.
Right. That's just pumping out antibodies.
Just pumping them out.
Okay.
And then importantly, you also generate memory B cells.
Right.
And those are really important for long-term immunological memory.
Right. So if you see that pathogen again, you've got that rapid response ready to go.
Much faster, much stronger response the second time around.
Okay. So then finally, let's talk about NK cells.
Okay.
So NK cells, they're lymphocytes,
Right.
but they act more like innate immune cells, right?
They do, yeah.
Yeah.
They provide that very rapid cytotoxic response,
Yeah.
but they don't need to be previously sensitized to an antigen.
Right. They're not specific.
Right.
Like a T cell is.
Exactly. And similar to CD8 T cells, they have these cytotoxic granules filled with perforin and granzymes.
Okay.
And they release those to kill infected cells or tumor cells. They also play a role in ADCC.
Antibody-dependent cell-mediated cytotoxicity.
Yes, exactly. So they can recognize cells that are coated with antibodies, and then they kill them.
Got it.
And then they also secrete cytokines,
Okay.
things like interferon gamma and TNF-alpha,
Mm-hmm.
which help to modulate the immune response.
Got it.
And they're regulated by a really interesting balance of signals.
Okay.
So they have activating receptors and inhibitory receptors. And the inhibitory receptors typically recognize MHC class I molecules,
Right.
which are present on most healthy cells.
Right.
So that basically prevents the NK cell from killing a healthy cell.
Okay.
But a lot of virus-infected cells, or tumor cells, they downregulate MHC class I.
Got it.
So they're trying to hide.
Right, right.
But that makes them susceptible to NK cell killing.
Yeah, so it's kind of like a fail-safe.
Exactly.
If they try to hide, the NK cells can still find them.
Yeah.
Okay. So we've got all these different types of leukocytes circulating in the blood, but they can't just, wander around aimlessly, right?
Right.
They have to get to where they need to go.
Yeah, that's where leukocyte extravasation comes in.
Right, so they're leaving the blood.
Right, they're leaving the bloodstream, and they're going into the tissues where they're needed.
Yeah.
And it's a very tightly controlled process.
Yeah, how does that work?
So the first step is rolling,
Okay.
where the leukocytes are kind of just loosely binding to the endothelium, and that's mediated by selectins.
Selectins, okay.
And then they're activated by chemokines that are released at the site of inflammation.
Right.
And that activation actually increases the affinity of integrins on the leukocyte surface.
Integrins, okay.
And then you get firm adhesion, where those integrins bind very tightly to adhesion molecules on the endothelium.
Okay.
So things like ICAMs and VCAMs.
Got it.
And then finally you get transmigration or diapedesis,
Okay.
which is where that leukocyte actually squeezes between the endothelial cells
Right, right.
and enters the tissue.
And actually, it's interesting you brought up adhesion molecules, right?
Yes.
Because if there's a defect in adhesion molecules,
Right.
then you have problems.
Absolutely.
Like leukocyte adhesion deficiency.
Yeah, LAD.
Yeah.
That's a very serious immunodeficiency.
Right, exactly. Okay, so the cells can get to where they need to go.
Yeah.
But then once they're there, we don't want them just, going crazy. They
Yeah.
need to be tightly regulated.
Absolutely, yeah.
Yeah.
Their activity has to be very tightly controlled.
So how does that happen?
So there's a lot of different mechanisms that play a role.
Okay.
Cytokines are really important.
Yeah.
You have different interleukins and interferons and the TNF and chemokines.
Okay.
That can either enhance or suppress leukocyte function.
Got it.
And then the complement system, beyond just directly killing pathogens, it also generates fragments that can attract and activate leukocytes.
Okay.
And then you have lipid mediators, things like leukotrienes and prostaglandins,
Right.
that are important for regulating inflammation and leukocyte recruitment.
Yeah.
And then you also have direct cell-cell interactions.
Okay.
So different immune cells can actually interact with each other through receptor ligand binding.
Right.
And that can either activate or inhibit their function.
So it's like they're constantly talking to each other
Absolutely.
and figuring out what to do.
Yeah, they're constantly sending signals back and forth.
Right. Okay, so just like with leukocyte production, things can also go wrong with leukocyte function, right?
Absolutely, yeah. And that can lead to a variety of clinical problems.
So what are we talking about there?
So you can have quantitative defects.
Yeah.
So that's where you either have too few leukocytes, like in leukopenia,
Right.
or you have too many leukocytes, like in leukocytosis.
Yeah.
But sometimes, even if you have a lot of leukocytes, they may not be functioning properly.
Right, they're just there, but they're not doing their job.
Exactly.
Okay.
And then you have qualitative defects, which is where the leukocytes are present,
Yeah.
but they're not working properly.
Right.
And that can be due to a variety of different things.
Okay.
You could have phagocytic disorders, like chronic granulomatous disease, or CGD.
Right.
And that's where neutrophils and macrophages have a defect in the NADPH oxidase enzyme.
Okay.
So they can't generate reactive oxygen species.
Right.
And they can't kill pathogens effectively.
Got it.
You could have leukocyte adhesion deficiency,
Yeah.
which we talked about, where they can't get to the site of infection
Right, right.
because they have defects in those integrins.
Yeah.
There could be defects in chemotaxis, degranulation, cytokine production,
Right.
all kinds of things.
Right, so any part of that process
Any part of that process can go wrong
can be messed up.
and lead to clinical disease.
Right, okay. So we've talked about how leukocytes are made, we've talked about their functions, but these cells don't live forever, right?
They don't, no.
So how are they cleared out? How are they destroyed?
So that's a very important process, leukocyte destruction. It's tightly regulated.
Yeah.
And it's really essential for maintaining immune homeostasis.
Right, so we don't want these cells just hanging around.
Exactly. We don't want them to accumulate.
Right.
We don't want them to become dysfunctional.
Yeah.
We need to get rid of them in a controlled way.
Right.
And the primary mechanism for that is apoptosis,
Apoptosis, okay. So programmed cell death.
programmed cell death. It's a very organized, very controlled process.
Okay, so how does that work in leukocytes?
So there's two main signaling pathways that can initiate apoptosis in leukocytes.
Okay.
You have the intrinsic pathway, which is also called the mitochondrial pathway,
Okay.
and that's triggered by all kinds of intracellular stresses.
Okay.
So things like DNA damage, nutrient deprivation, if they're not getting enough survival signals from growth factors,
Got it.
all of those things
Mhm.
can trigger the intrinsic pathway.
Okay.
And that leads to changes in the mitochondrial membrane and the release of cytochrome C,
Okay.
which then activates a cascade of caspases.
Right.
And caspases are really the executioners of apoptosis.
Right. Right.
And then you have the extrinsic pathway, which is also called the death receptor pathway.
Oh, okay.
And that's initiated by the binding of death ligands.
Okay.
So things like Fas ligand or TNF alpha
Right.
to their death receptors on the surface of the cell.
Got it.
So things like the Fas receptor or
Wow.
TNF receptor.
Right.
And that binding directly activates caspases.
So both pathways converge on the caspases.
Absolutely. Yeah, both pathways ultimately lead to caspase activation.
Okay.